Hello everyone, welcome to Elite Rankers Academy. This is your mentor Dr. Shamaita Basu, and in this session we will start with the introductory part of biomolecules. So starting with the introduction before we go on into the major concept or the major details of biomolecules, because biomolecules are those molecules which make up our cell, okay, and that's why in continuation with the introductory part of biomolecules we will go to the details of the structure and functions of the cell. So let's first just focus on the introductory part. So whenever we are talking about our biosphere, okay, now what do you actually mean by biosphere over here? So just let's focus on this diagram. So here biosphere basically consists of three things: water, air and soil. Okay, just try to remember it like that. Now the good name for water will be hydrosphere. Hydro means water. The good name for air will be atmosphere, and the good name for soil will be lithosphere. Okay, so these three things will be making up our biosphere, and inside the biosphere, obviously, we, the biological living beings, the humans, the animals, the plants, they also will belong in this biosphere only. Okay, so I'm just repeating, the three main parts of biosphere will be water, air, and soil. Okay. Okay, now moving forward, now there is a question which can come in our mind, because uh, like interactive sessions are very much helpful to help you understand a thing in details and to focus on the major conceptual parts of these things. Okay, because you are the students who are preparing for NEET. You are preparing to appear for NEET, right? So NEET is a competitive exam, you all know, and it is having negative marking. So it's very important that you can grab the important parts and the conceptual parts which will help you eventually to solve the different MCQs that you will be getting in the final exam. Okay. So after I complete each and every topic, we will be discussing several MCQs which will be relevant or important pertaining to that particular topic. Okay. So the first question that is coming in our mind as far as biomolecules are concerned is that what are the chemicals that the living system is made up of? Living system means the living organism, us humans. Okay, so there will be elements and there will be compounds. Now the question is whether all the living organisms are made up of those same elements and compounds. Okay, now from here an important concept again arises, that is elemental analysis. Okay, now what is elemental analysis? Analysis means when you are analyzing something. Okay, you are trying to find out a pattern, okay, or a reason for something. That is analysis. Elemental means when you are analyzing the elements. What are the elements that are present in the living organism? Okay, so moving forward, what is actually elemental analysis, which I already made you understand in a much simpler process. So it is a process where a sample of some material which will be containing some type of elements, obviously. Okay, now those elements can be in the form of minerals, they can be in the form of organic compounds. Okay, and the material or the sample that we are talking about, it can be soil, it can be waste, it can be drinking water, it can be any body fluid. Okay, any body fluid, so for example saliva, okay, sweat, that is body fluid. Any mineral, okay, minerals example like zinc, calcium, potassium, magnesium, these are various important minerals which are present or which form an important part of the living body. Okay, and different chemical compounds. So that is basically the how will you define elemental analysis. Okay, now again something that I would like to mention over here. Now these definitions or these like it's not important that you have to memorize, you have to mug up. Okay, it's not important. But what is important is that you have to know the concept behind that definition. You have to know the reason. Okay, then only you will be able to solve the MCQs, because in MCQs you just need to pick the right option, isn't it? You just need to circle the right option in the OMR sheet. That is exactly what you need to do. not write to you do not need to write the answers, but in order to tick or in order to select that particular right option, these are the definitions that you need to remember. Okay, these are the components, these are the concepts that you have to be clear in your mind. Okay. Anyways, moving forward. So when elemental analysis is performed on a plant tissue, animal tissue, or a microbial paste. Okay. Now see, plant tissue means coming from a plant, obviously a living thing. Animal tissue means coming from an animal or a human being, again a living thing. And microbial paste means a component or a part which has been obtained from a microorganism. Microorganism में क्या क्या आएगा? Bacteria आएगा, fungus आएगा, parasite, protozoa and virus. Okay. So ये जितने भी microorganisms है, they are also living beings, isn't it? So here we are talking about elemental analysis of living beings. So there will be a list of elements which will be obtained from each of this living tissue. Okay, what are the list of elements? Let's see. I have just marked it with red. Okay, so that it's easily visual to your eyes. So carbon, hydrogen, oxygen. Now here comes a very important concept, that carbon, hydrogen and oxygen, these three elements, they form the organic compounds in our body. Okay, and as a living organism or as a human being to be precise, what are we? We are nothing but made up of organic compounds. In our body, 80 to 90% compounds will be organic compounds. Okay, basically all the compounds will be organic compounds. Then only we will be able to be a living organism. Okay, and the major elements of these organic compounds will be either carbon or hydrogen or oxygen. So this is a very important concept that you need to remember. Okay. So now, if we are doing a same kind of analysis, same kind of elemental analysis using two samples, then we are obtaining a similar list. Okay, two sample means इसमें एक तो already living tissue था, which is either plant tissue, animal tissue or microbial paste, and the other sample that we are taking, say for example it's a piece of earth's crust. Piece of earth's crust मतलब क्या? उसमें ज़्यादा से ज़्यादा soil रहेगा, means a component of the lithosphere. फिर पानी रहेगा, that is a component of the hydrosphere, isn't it? ये दोनों component mainly रहेगा. So they are non-living tissue, isn't it? तो इसका भी अगर हम elemental analysis करेंगे तो उसमें भी we will getting a similar type of list. Okay, now ये दोनों list में difference क्या होगा? Obviously कुछ ना कुछ difference तो होगा ही, है कि नहीं? तभी अलग अलग सैंपल का हम लोग एनालिसिस करेंगे. अगर कोई भी डिफरेंस नहीं निकालता तो अलग अलग एनालिसिस करने का कोई मतलब ही नहीं होता, isn't it? Now what are the difference between the two lists? अगर हम एब्सोल्यूट टर्म्स पे देखें, एब्सोल्यूट मतलब ऊपर ऊपर, ठीक है, ऊपर ऊपर से अगर देखें तो कोई भी apparent differences will not be visible. बट अगर हम गहराई में जाके देखेंगे, ठीक है, अगर एकदम प्रिसाइसली देखेंगे तो देखेंगे कि आज क्रस्ट में जो एलिमेंट्स था वो लिविंग टिश्यू के एलिमेंट्स में भी है, मतलब दे आर द सेम. ऑल द एलिमेंट्स जो कि आज क्रस्ट के सैंपल में था, दे आर ऑल्सो प्रेजेंट इन द सैंपल ऑफ द लिविंग टिश्यू. तो अभी तक कोई बेसिक और ग्रॉस डिफरेंस नहीं मिला है. लेकिन जब हम गहराई में जाएंगे, जो यहाँ पे बोल रहा है क्लोजर एग्जामिनेशन, क्लोजर एग्जामिनेशन करेंगे तो क्या देखेंगे कि कार्बन एंड हाइड्रोजन का जो कंपोनेंट है, कार्बन एंड हाइड्रोजन का जो अमाउंट है, वो लिविंग टिश्यू वाला सैंपल में ज्यादा मिलेगा देन इन कंपैरिजन टू द सैंपल ऑफ द अर्थ क्रस्ट. Okay, so that is exactly the explanation of the thing that I mentioned sometime before. कि in the living organism, in the human beings or any living tissue, the component or the the content of organic compounds like hydrogen, oxygen and carbon will be much more. ठीक है, moving forward. So again the same thing they are showing in the form of a table. What are the, how will you compare the different elements with respect to whether it's a non-living tissue or it's a living tissue. Okay, so just let's see the table over here. As you can see, कि left hand side में there are the different elements and right hand side में एक ही column को two parts में divide किया गया है, जिसमें एक में है earth's crust और एक में है human body. Okay, So now just see hydrogen, carbon, oxygen, as I mentioned, they have to be present in a more percentage in the human body since they are forming the organic compounds. So just see exactly what I said. In Earth's crust, percentage of hydrogen will be 0.14. In human body, percentage of hydrogen will be 0.5. Okay. Now, what do you mean by percentage weight? Percentage weight means per kg or per gram. ये percentage होगा 0.14% per kg. ठीक है. तो if a human body, if the weight of that person, male or female, whatever, is 50 kg, then उसमें 0.5% per 50 kg will be the unit. Okay. I think I have made myself clear. So that is for hydrogen. Next coming to carbon. Carbon में भी same चीज है देखो. Earth's crust में only 0.03%, human body में 18.5%. So it's multiple times. Okay, I think more than 5 or 6 times multiplied. Next is oxygen. Again the same thing. 46.6% in Earth's crust. Human body में it's becoming 65. Next moving on to nitrogen. Nitrogen again is a very important component of each and every living being. Okay, why? Because from nitrogen we get the amino acids, and from amino acids we get the proteins. Okay, these are some of the things which I will be explaining in much more details in the upcoming sessions of biomolecules. We have just begun with the introductory part, so I just don't want you to confuse, but just remember that nitrogen is also a very important component of the living being. Okay, whether it's plant, animal or human. So नाइट्रोजन जो है अर्थ क्रस्ट में बहुत ही कम होगा, ह्यूमन बॉडी में 3.3% होगा. Sulfur will be 0.03 in Earth's crust, human body 0.3. Sodium will be 2.8 in Earth's crust, human body में 0.2. So I think sodium is the first mineral जो हम देख रहे हैं, या first element जो हम देख रहे हैं कि Earth's crust में ज़्यादा है in comparison to human body. Okay. Calcium again 3.6 in Earth's crust, human body 1.5. लेकिन इसका मतलब ये नहीं है कि सोडियम, कैल्शियम ये सब इंपॉर्टेंट मिनरल्स नहीं है, ये भी बहुत ही इंपॉर्टेंट है फॉर द ह्यूमन बॉडी. Okay. So for example, calcium is very important for the bones. Sodium is very important electrolyte which must be present in the body to maintain the normal pH. Okay. Next is magnesium. Magnesium Earth's crust में 2.1, human body में 0.1, again less. Silicon is something which is almost not present in the human body, but Earth's crust में percentage will be 27.7. Moving on to the next slide. Now, how will you actually analyze the chemical composition? ये जो elemental analysis के बारे में हम लोग बोल रहे हैं, वो actually में करेंगे कैसे? Okay, how will you perform it in the lab, say for example? So, इसमें भी एक सवाल उठता है कि what type of organic compounds are found in the living organisms? ये जो इतना हम बोल रहे हैं organic compounds, what are the type? And how will you get the answer? Obviously by performing the chemical analysis of those organic compounds. Okay, so here we will be focusing on that only. So firstly we will be talking about chemical analysis, and then we will be talking about the composition. Okay, so chemical analysis is basically the study of the chemical composition and structure of substances. Okay, say for example I have got a chunk of tissue, living tissue, okay, from anywhere, say it's a plant tissue or animal tissue, any type of living tissue. तो उसमें what are the chemicals that are present, उसका composition क्या है, and depending on that what is the structure of that tissue, that is what is known as chemical analysis. Okay, so we can take any living tissue for that. Exactly what I said, we can take a piece of a vegetable, we can take a piece of a liver. Okay, and then what we need to do is we need to have a mortar and pestle. Okay, what is mortar and pestle? It's basically a grinding technology. It's used for grinding. So you have a container or a katori type looking, and then you have a a cylindrical piece of usually it's made up of marble. Okay, both the katori material and that पीसने वाला जो सामान होगा, both of them ideally made up of marble. Okay, so you just have to put whatever thing you are trying to grind, जो भी पीसना है उसको अंदर डालो, कटोरी वाला जो सामान है उसमें, that is also made up of marble. Okay, और जो cylindrical पीस है जिसको हम पेसल बोलेंगे, उससे अच्छे से उसको पीसना है. Okay, so that is mortar and pestle. तो उसमें डालना क्या है, पहले तो ऑब्वियसली यू हैव टू पुट योर सैंपल विच कैन बी अ वेजिटेबल और इट कैन बी अ पीस ऑफ liver, whatever it is, and उसमें you have to put trichloroacetic acid and the formula of trichloroacetic acid will be Cl3CCOOH. Okay, formula is again something important as far as your exam NEET is concerned, because in many questions they just ask the formula. Okay, so then after that, पीसने के बाद आफ्टर दैट ग्राइंडिंग व्हाट यू विल गेट अ थिक स्लरी के एक मतलब एक लिक्विड टाइप का चीज मिलेगा, क्योंकि एसिड डाला है, जो कि बहुत ही ठीक होगा। ओके, उतना भी मतलब पानी जैसा लिक्विड भी नहीं होगा। ओके, एंड इफ वी वेयर टू स्ट्रेन दिस थ्रू अ चीज क्लॉथ और कॉटन, तो उसके बाद एक कॉटन, अच्छा सा साफ कॉटन का पीस लेना होगा और उसमें उसको छन्ना होगा। okay, छन्नी का जो काम है बेसिकली वही कर रहा है थ्रू द कॉटन। ओके, so after that straining part, filtration part, we will be getting two types of filtrate. Okay, एक को बोलेंगे acid soluble filtrate और acid soluble pool, और दूसरे को बोलेंगे acid insoluble fraction और acid insoluble pool, जिसका दूसरा नाम होगा retentate. Okay, so these are two important terms that you need to remember, so I will just highlight it for you. So one is acid soluble pool and the other one is acid insoluble fraction or retentate. Okay, तो बेसिकली क्या हुआ था कि हमने एसिड डाला था, तो एसिड डालने के बाद जरूरी नहीं है कि जो भी है सब कुछ घुल जाएगा एसिड में, है ना। तो कुछ कुछ सामान होगा जो घुल जाएगा, जो सॉल्युबल हो जाएगा, और कुछ कुछ सामान होगा जो इनसॉल्युबल रहेगा। तो whatever gets solubilized will be known as the acid soluble fraction and whatever is remaining insolubilized will be known as the acid insoluble fraction. Okay, तो ज्यादा से ज्यादा ऑर्गेनिक कंपाउंड्स जो होगा वो हमें मिलेगा कहाँ पे, लेट्स सी हाउ डज केमिकल कंपाउंड्स आर एनालाइज। सो बेसिकली ये जो लास्ट स्लाइड में मैं बोली थी उसी का एक फ्लो चार्ट वाला वर्जन इसमें दिया हुआ है, बिकॉज यू नो whenever you are studying something new, okay, because you know many topics, some of the topics it's not necessary that each and every topic will be interesting for you. Okay, because obviously whoever you are coming to prepare for NEET, obviously you have come because you have a love for that subject, isn't it? You have a love for biology, you have a love for zoology and botany, that's why you have come to prepare for this exam. लेकिन उसमें भी कभी कभी क्या होता है कि जो सब्जेक्ट हम पसंद करते हैं उसमें भी कुछ कुछ ऐसा टॉपिक्स होता है जो बोरिंग बन जाता है। ओके, तो इसलिए जैसे कि पर्टिकुलरली फॉर मी बायोमोलिक्यूल्स, जब like when I used to study all these topics मेरे बचपन में, this topic was actually a boring topic for me. Okay, so whenever some things are getting boring, it's important that, like I'm just giving a suggestion, it's important that you make flow chart, you make tables, you make nice diagrams. Okay, or you make a nice a pie chart. Okay, so that and try to make it colorful. Okay, क्योंकि जितना भी हमारा ब्रेन जो है ना वो बहुत बेवकूफ है, उसको हम जैसे मतलब वो करेंगे, प्रोसेस करेंगे, वो वैसा ही सोचेगा। तो विजुअली अपीलिंग थिंग्स जो है, मतलब जो दिखने में अच्छा लगता है, कि जो भी चीज तुम कलरफुल देखोगे वो दिखने में अच्छा लगेगा, तो वो चीज ब्रेन में ज्यादा देर तक रिटेन होगा। ओके, वो इंफॉर्मेशन विल स्टे फॉर अ लॉन्गर टाइम इन द ब्रेन इफ इट्स कलरफुल, इफ इट्स नाइसली अरेंज्ड, इफ इट्स इन द फॉर्म ऑफ अ नाइस फ्लो चार्ट, then your brain will automatically remember it for a longer time. Okay, so that's why I have tried to put the slides like that. ओके, सो लिविंग टिश्यू लिया पहले, उसमें वेजिटेबल भी हो सकता है और पीस ऑफ लीवर हो सकता है, मतलब बेसिकली एनी काइंड ऑफ लिविंग टिश्यू। उसके साथ वी आर मिक्सिंग ट्राइक्लोरोएसिटिक एसिड, देन वी आर ग्राइंडिंग इट इन मोर्टर पेसल, आफ्टर दैट वी आर obtaining a thick slurry, and that thick slurry is then filtered through a cotton cloth, and after filtration we are getting an acid soluble fraction which is known as the filtrate and a acid insoluble fraction which is known as the retentate. Now acid soluble fraction का definition क्या होगा? What is it? जो acid में soluble होगा, सेल का जो कंपोनेंट एसिड में soluble हो जाएगा उसको हम acid soluble fraction बोलेंगे। वेरी इजी। नाउ देयर आर, जो मैं पहले भी बोली थी कि ज्यादा से ज्यादा ऑर्गेनिक कंपाउंड्स जो है वो एसिड सॉल्युबल, तो वो हमें मिलेगा एसिड सॉल्युबल पूल में। अब आते हैं एसिड इनसॉल्युबल फ्रैक्शन किसको बोलेंगे, जिसका दूसरा नाम है रिटेंटेट। उसको कैसे डिफाइन करेंगे? जो एसिड में नहीं घुलेगा, जो एसिड में सॉल्युबल नहीं होगा, उसको हम बोलेंगे एसिड इनसॉल्युबल फ्रैक्शन। ठीक है, तो एसिड इनसॉल्युबल फ्रैक्शन जो होगा उसमें बस 4 टाइप का ऑर्गेनिक कंपाउंड्स होगा। क्या क्या? प्रोटीन, न्यूक्लिक एसिड, कार्बोहाइड्रेट एंड लिपिड्स। ओके, so this is something very important, because whenever we are talking about the human body, about the living system, these are only the main important organic compounds that are present. Okay, protein, carbohydrate, lipid, nucleic acid. हम सेल देख ले, न्यूक्लियस देख ले, डीएनए देख ले, कोई भी ऑर्गन देख ले, कोई भी टिश्यू देख ले, उसमें यही 4 कंपोनेंट्स मोर और लेस होता है, या तो प्रोटीन, या तो कार्बोहाइड्रेट, या तो लिपिड, या तो न्यूक्लिक एसिड। ओके, moving forward, now in higher classes, when you will be moving on to your graduation and then eventually post graduation, you will be learning a lot more about how to analyze in details these living tissues and how to actually identify a particular organic compound. Okay, so abhi tak itna hi bolna zaruri hai कि बहुत सारे separation techniques होता है to separate one compound from another. Okay, okay, now coming on to each of like, not details, you do not require details, but you at least you need to know the names. Okay, so firstly extraction. एक्सट्रैक्शन मतलब क्या? हम कुछ चीज एक्सट्रैक्ट कर रहे हैं, बाहर निकाल रहे हैं। ठीक है, सो इट्स एक्चुअली अ वेरी कॉमन लैबोरेटरी प्रोसीजर यूज्ड व्हेन आइसोलेटिंग और प्यूरिफाइंग अ कंपाउंड फ्रॉम इट्स मैट्रिक्स। मैट्रिक्स मतलब मेन जो उसका कंपोनेंट है उससे हमें अलग करना है, छोटा छोटा अलग अलग कंपाउंड्स। ओके, नेक्स्ट कमिंग टू सेपरेशन टेक्निक। सेपरेशन टेक्निक को कैसे डिफाइन करेंगे? मास ट्रांसफर फेनोमेना that converts a mixture of substances into or more distinct product mixtures which may be referred to as fractions। exactly जो हम पहले देख रहे थे कि एक एसिड soluble fraction मिलेगा, एक एसिड insoluble fraction मिलेगा। ठीक है, तो सेपरेशन के लिए what we do, we usually use a separatory funnel। okay, so in that separatory funnel, or through that separatory funnel, we are actually passing the solute. Okay, and then we are seeing the solute in the organic phase also, and then in the aqueous phase also. Okay, aqueous phase means more water will be there and less of the solute will be there, and organic phase means more solute will be there in comparison to the solvent fraction. Okay, I hope you know what is solute and solvent. Solute means which is getting soluble, and solvent is the medium which is helping the solute to get soluble. Okay, now separation techniques actually helps in separation of desired component from others. Okay, so say for example we have a mixture, so we will be having organic as well as inorganic compounds in that mixture, but if we want to only separate the organic compounds out from that mixture, then we need to go for the separation techniques. Now analytical technique basically deals with the methods for determining the chemical composition or structure of sample, means you are analyzing कि सिर्फ ऑर्गेनिक कंपाउंड है ये जानने से काफी नहीं है, कौन सा ऑर्गेनिक कंपाउंड है ये भी मेरे को जानना है, देन वी विल बी गोइंग फॉर एनालाइजिंग। ओके, सो एनालाइजिंग के लिए वी कैन गो फॉर क्रोमेटोग्राफी। ओके, क्रोमेटोग्राफी इज अ टेक्निक व्हिच इज क्रोम मींस कलर, ऑलवेज रिमेंबर क्रोम मींस कलर। सो इन क्रोमेटोग्राफी वी विल बी detecting or we will be analyzing the nature of the compound by the change in color. Okay, and change in color कैसे होगा? Depending on the density of that particular compound। जिसका डेंसिटी ज्यादा होगा वो सबसे पहले नीचे आएगा, उसका कलर चेंज होगा। स्पेक्ट्रोस्कोपी, because you know these methods you will be learning in much more details in the upcoming higher levels of your study. Okay, so till now or at this stage for your NEET you just need to know this much, not more than that. Okay, next important technique, analytical technique will be spectroscopy. Okay, spectroscopy क्यों आ रहा है? spectroscopy word आ रहा है from the term spectrum. Okay, तो स्पेक्ट्रम मीन्स क्या? स्पेक्ट्रम मीन्स एक ग्रेडियंट होगा जिसमें डिफरेंट कलर्स विल बी देयर। ओके, सो देयर विल बी अ मेन लाइट सोर्स, ओके, व्हिच विल बी scattering through a particular prism, and after getting scattered through that prism it will be splitted into different shades of color, as you will see. Okay, so depending on what color your compound is giving, you will be able to detect what nature of that compound is, or what amount and what nature of that compound is there. Okay, another important analytical technique will be NMR spectroscopy. Okay, where we use a machine. It's a totally machine oriented process. Even spectroscopy is a machine oriented process. You just need to put your sample inside and then automatically the machine will be giving you the reading. Okay. Manual spectroscopy are also there, but nowadays we do not use that. Okay. NMR spectroscopy may also, this is a totally machine or instrument oriented, so this is how it will look like. Okay, so you can have questions where this image will be given and maybe you will be having 4 options. So from there you will have to select NMR spectroscopy as the right option. Okay, NMR stands for nuclear magnetic resonance. Okay. So the analytical techniques, when they are applied to the compound, it gives the idea of the molecular formula or the probable structure of the compound. Okay. Say for example the molecular formula for glucose, it is C6H12O6. तो उसका formula निकालने के लिए, उसका particular structure निकालने के लिए, we will go for this analytical techniques. Okay. Now coming to the main topic of this session, that is biomolecules. So biomolecules are basically the carbon compounds which are obtained from the living tissues. Okay. Carbon, as I said, is a very important organic element present in the living system. Okay. But it doesn't mean that it only has the organic compounds, it has inorganic elements and compounds as well. Okay. So talking about analysis of inorganic elements of living tissue. So I think this is somewhat that I told in the previous slides where we talked about acid soluble pool and acid insoluble fraction. So इसमें basically क्या हो रहा है, उसमें हम लोग organic compounds analyze कर रहे थे. इसमें we are mainly focusing on the inorganic elements. So again we are taking some type of living tissue which is either a leaf plant tissue or animal tissue. Then we are just drying the tissue. So after drying, what we will be getting, a dried living tissue. Then we will burn the tissue at high temperature. After burning, what will be happening? Whatever carbon compounds are present in that living tissue, they will be getting oxidized to the gaseous form, that is CO2, carbon dioxide and water vapor. So that is the utility behind burning. Okay, any carbon compound को burn करेंगे तो उससे CO2 and water vapor मिलेगा. So that is basically the ash material, जिसको हम छाई बोलते हैं. Ash मिलेगा. Okay, and in this ash there will be various inorganic components or elements present like calcium and magnesium. So this is just a picture of how that ash will look like, and in this ash the major elements that we will be having will be calcium and magnesium. So inorganic compounds like sulfate, phosphate will also be seen in the acid soluble fraction, which I already talked about. Okay. And elemental analysis will be giving us an elemental composition of living tissues in the form of hydrogen, oxygen, fluorine, carbon. Means what are the elements present in the living system, उसके बारे में हमें जानकारी मिलेगी. While analysis of compounds gives an idea of the kind of organic and inorganic constituents present in the living tissue. Okay, so one is elemental analysis, one is compound analysis. Okay, compound analysis करेंगे तो कौन सा type of organic या inorganic है वो पता चलेगा, और अगर elemental करेंगे तो what are the different elements वो पता चलेगा. Okay. So from a chemistry point of view, अगर हम chemistry के जरिए से देखेंगे तो हम different जो functional groups है इसको भी हम identify कर पाएंगे, like aldehydes, ketones and aromatic. Okay, these are again something that you have already either learned or you will be learning in the upcoming classes of your chemistry part of NEET. Okay. और अगर बायोलॉजिकल पॉइंट ऑफ व्यू से देखें जो हमारे लिए यहाँ पे इम्पोर्टेंट है, then we will be able to classify them into proteins, fats, lipids and nucleic acids. Okay, so this is exactly how the different aromatic compounds will look like. Again, this is important for your chemistry part. Okay, so talking about amino acids, if we talk about amino acids, as I mentioned, the major important component of amino acid will be nitrogen. So this is the nitrogen part of the N terminus, and this is the carbon part, that is the C terminus. Okay, and this is the where we will be having the functional group. So depending on the functional group, each type of amino acid will vary. Okay, and there are a group of essential amino acids, there are a group of non-essential amino acids. Okay, total there are 21 amino acids that are there or that are important for your body for making the proteins. Okay, so how those 21 amino acids will be formed? डिपेंडिंग ऑन दिस R group, दैट इज द फंक्शनल ग्रुप. जैसे ही R group चेंज होता जाएगा वैसे वैसे एक नया नया अमीनो एसिड हमें मिलेगा. Okay, so I think over here again we have a table type of a format where they are giving different important inorganic constituents and their respective formula. So sodium के लिए Na plus we all know, potassium के लिए K plus, calcium के लिए Ca2 plus, magnesium के लिए Mg2 plus, water के लिए H2O and यही सबका जब कंपोनेंट बनेगा तब क्या होगा, से फॉर एग्जांपल सोडियम के साथ क्लोरीन, सो NaCl होगा. कैल्शियम के साथ CO3, कैल्शियम कार्बोनेट होगा. देन PO4 माइनस मतलब फॉस्फेट, SO4 माइनस मतलब सल्फेट. सो दीज आर द इम्पोर्टेंट कंपाउंड्स. ओके. Now, last but not the least, as I already mentioned in the beginning of this session, that we will be talking about important MCQs relevant to that particular session. Okay, तो इस session का अगर बात करेंगे तो first MCQ जो दिख रहा है देखो, the study of the chemical composition and structure of substances is called chemical bonding, chemical synthesis, next option, chemical analysis, next option, and next is none of this. Okay, so obviously the right option will be chemical analysis. Next important MCQ that we have, inorganic element present in ash. क्या क्या था, कैल्शियम था और मैग्नीशियम था. ओके, तो कैल्शियम ऑप्शन है नहीं, ओनली मैग्नीशियम इज द ऑप्शन दैट इज प्रेजेंट. सो ऑब्वियसली द राइट ऑप्शन विल बी मैग्नीशियम. MCQ में the main thing that I need to or that I want to tell you is that whenever you are solving the MCQ, it's very important that you read the question very carefully and it's very important that you read each of the options very carefully. Okay, because बहुत बार क्या होता है, क्वेश्चन पढ़ के ही हमें लगता है कि अरे ये तो बहुत आसान है, उसके बाद क्या होता है जो ऑप्शंस है वो हम अच्छे से देखते ही नहीं है. ठीक है, जैसे ऊपर ऊपर ऑप्शंस देखते हैं ये राइट है. ओके, सो दैट इज द रिजल्ट ऑफ ओवर कॉन्फिडेंस एंड ओवर कॉन्फिडेंस इज लीथल, इट इज हार्मफुल. तो ओवर कॉन्फिडेंट नहीं होना है. जितना भी क्वेश्चन आसान लगे उसको अच्छे से पढ़ना है, एटलीस्ट 1 से 2 बार उसको रिपीट करना है, उसके बाद ही राइट ऑप्शन टिक करना है. बिकॉज वंस यू सर्कल दैट दैट स्पेस इन द OMR शीट, देयर इज नो अदर ऑप्शन, यू कांट चेंज इट, इजंट इट, यू कांट अनडू इट. तो एक ही बार में करना है, फाइनल करना है. ठीक है, और बहुत सारे केसेस में क्या होता है कि इन द क्वेश्चन ओनली द आंसर इज गिवन इनडायरेक्टली. इन मेनी क्वेश्चंस आर लाइक दैट, चूज the option that is not correct, incorrect नहीं लिख के option that is not correct ऐसे करके देता है. तो उसमें क्या होता है, it's just to confuse you people, nothing else, confuse करने के लिए. ठीक है, तो इसीलिए it's important that you read the question very carefully and in many cases what happens, the options are very confusing. ओके, फर्स्ट टाइम जब पढ़ोगे ऐसा लगेगा कि अरे सारे के सारे ऑप्शंस राइट है, कौन सा चूज करें इसमें से. तो इसीलिए इंपॉर्टेंट है कि एक क्वेश्चन को एटलीस्ट दोबारा या 3 बार पढ़ा जाए. ओके, उतना टाइम रहता नहीं है, दैट इज समथिंग आई अंडरस्टैंड, बट स्टिल यू हैव टू डू दैट. That is only time management and for that only these assignments, these assessment tests are done. Okay, so next is which of the following elements abundance is more in any living organism than in Earth's crust. Okay, carbon, hydrogen, sodium and magnesium. Right option क्या होगा, option A and B. Okay, तो इसमें actually एक option ऐसा होना था, both A and B. Okay, because A and B is actually not an option from any of these 4 options. So one of the options should be both A and B, क्योंकि carbon, hydrogen and oxygen, these are the 3 main organic compounds जो मिलता है. Okay. Okay, sorry, very sorry. These are the 4 options that we have. Among them, the right option will be, देखो यहाँ पे हाईलाइट किया है abundance. What is abundance? Abundance मतलब जो सबसे ज्यादा होगा. ठीक है, so the right option will be A and B. Okay. Next is which of the following techniques when applied to the compound gives us an idea of the molecular formula and probable structure of the compound. What are the options? Analytical techniques, separation techniques, both analytical and separation techniques, and then none. Okay, तो इसका सही ऑप्शन क्या होगा, इसमें भी देखो इंपॉर्टेंट जो पार्ट्स होगा, इसलिए मैं बोल रही हूँ कि क्वेश्चन पढ़ना बहुत इंपॉर्टेंट है. क्योंकि क्वेश्चन में बहुत सारा वर्ड लिखा होगा, इसमें से इंपॉर्टेंट पार्ट तुमको चुनना है, तुमको देखना है, देन ओनली यू विल बी एबल टू गेट द राइट आंसर. ओके, तो इसमें What will be the right answer? Analytical techniques, क्योंकि molecular formula and probable structure दोनों के बारे में बोले, ठीक है. Next is chemical analysis of living tissue can be obtained by using what chemical? Trichloroacetic acid, trichloroethylene, trichloric acid, and trichloromethane. Obviously, the right answer will be trichloroacetic acid. Okay. So I think that's it about this session. We will be soon moving on to the next session regarding biomolecules. Thank you and goodbye.